Hello everyone. I would like to present our project research titled Applying Autoencoders of Molecular Evolution of Coronaviruses. This project is a result of 6-month research of the following students, me and Ming. As you may all hear about the word coronavirus, this is actually a group of related RNA viruses that cause disease in mammals and birds. In humans and birds, they cause respiratory tract infection that can range from mild to lethal. The more lethal varieties can cause SARS, MERS, and COVID-19, which is one of the most dangerous epidemics in the recent history. In combating against those viruses, phylogenetic analysis is very useful. First of all, we can use molecular genetic data to reconstruct the transmission tree, such as procedure can use molecular genetic data to reconstruct the transmission tree, such as providing a mean to complement traditional method of contacting tracing. In tradition, insight based on the phylogenetic relationship among the coronaviruses is also helpful to discover drugs and develop vehicles against the viruses. After reviewing scientific literature, we propose a new method for phylogenetic data of coronavirus, especially of SARS-CoV-2, utilizing an alignment-free approach in combining with a machine learning technique to classify the viral genomes.
We carry out our research in the following steps. To begin, we collected all known 92 coronaviruses genomes from the RefSeq database, from the GenBank collections of NCBI. We also collected over 14,000 genomes of SARS-CoV-2 from the GSAID database, an initiative that promotes the rapid sharing of data from all influenza viruses and the coronaviruses causing COVID-19. The second step is processing of this genomic data by calculating the nucleotide frequencies, word count, or chimer frequencies of the genomes. This step aims to compare the genomes in whole based on an alignment-free approach, and this approach has been proved to offer several advantages compared to the classical approach of multiple alignment, such as carrying out more quickly, does not depend on a certain evolutionary module, can detect rearrangement or horizontal transfer events. And these pictures illustrates the principle of the word count or chimer frequency calculations. For the certain chimer size, for example 3 here, we count the numbers of all word size 3 with the step of one nucleotide along the sequence at a time.
With this research process, we observed that k value equal to 11 offer the best performance in terms of classification of genomes in speed and in agreement with the virogeny. Using that value, we obtain two main resources as follow. First, we can use a phylogeny for all known coronavirus very quickly in this graph. This tree can separate correctly in the 4 main groups of coronavirus, which are alpha, beta, delta, and gamma. This confirms the value of our approach. In addition, by taking the consideration all known coronavirus, our results offer a few surprising information. For example, such as SARS-CoV-2 was grouped most closely to RAS coronavirus. This information is worth checking and may offer a new information about the source of that dangerous virus. Secondly, we apply our approach to over 14,000 known SARS-CoV-2 genomes and saw them classified 3 main groups or subfamily based mostly on the geographical source of the viruses. The subgroup A contained most of the strain from Asia and showed a long time of deviation, a proof of a long evolution time in the region. A subgroup B contains most of the strain from the Europe and showed close relationship, a proof of a shorter evolution time. The subgroup C contains the strain from the rest of the world and has the diversity of the strain without any clear-cut grouping. It shows a diversity of the sources of the viruses around the world.
Over the 20 species of coronaviruses that we have analyzed, we discovered that the virus has 3 main subfamilies, which is based mostly on geographical distribution, and that most of genetically similar viruses tend to group according to the evolution time. In conclusion, the combination of a new deep learning method called Autoencoder with the alignment-free method to compare the whole virus genomes can build a full phylogenetic relationship of the viruses, which is in agreement with the classical methods in the main points, but offer more useful insights about the source of human-infected coronaviruses.
We plan to optimize further the autoencoder model so that we can analyze better the viral genomes. And we shall apply the method to identify the original host and the emergent direction of SARS-CoV-2, especially of the strain in Vietnam.